Welcome to this explainer. Today, we're going to dive into the elite, microscopic manufacturing process that's happening literally millions of times a second inside your very own cells. We're talking about eukaryotic transcription. Think of your body like an incredibly complex metropolis. For it to function, billions of tiny factories need to constantly read the original blueprints of life, your DNA, and manufacture the exact parts required to keep you alive and thriving. So we're going to step inside one of those factories and decode exactly how this microscopic assembly line operates.
Before we start throwing acronyms around, let's just establish a quick baseline. Eukaryotic transcription is the highly regulated procedure where your cells copy DNA into RNA. And a really vital piece of this definition to keep in mind is that this all happens securely within a membrane-bound nucleus. You see, your precious DNA, it never actually leaves this protective vault. Instead, the cell creates a copy, an RNA transcript that can safely be sent out to the wider cellular factory.
Okay, here is our blueprint for today. We'll start with part 1, the cellular assembly line. Then we'll move through stage 1, initiation, stage 2, elongation, and stage 3, termination. Finally, we'll wrap things up with part 5, eukaryotes versus prokaryotes.
Part 1, the cellular assembly line. To keep us from getting totally lost in the biological jargon, just remember that the heavy machinery in our cells always follows these 3 strict chronological steps. It makes perfect sense, really. Step 1 is initiation, getting the machine started. Step 2 is elongation, actually running the machine to build the product. And step 3 is termination, stopping the machine and releasing that finished product. Basically, everything we talk about today is going to fit neatly right into one of those 3 buckets.
Moving on to part 2, stage 1, initiation.
So our heavy machinery, specifically this enzyme called RNA polymerase II, well, it can't just attach anywhere on the DNA all by itself. It's super powerful, but it's kind of blind. It needs help. That's where general transcription factors, like TFID, come in to act as the managers. They scan the DNA strand looking for specific core promoter sequences, like the famously named TATA box. Once they find it, they bind right to the DNA, signaling the exact spot where construction needs to begin. This builds a pre-initiation complex that recruits our RNA polymerase II. Finally, they literally melt or unwind the DNA strands, exposing the precise template that needs to be copied. And boom, the assembly line is powered up and ready to go.
Next up is part 3, stage 2, elongation.
Now that everything is set up, the machinery needs a chemical green light to actually hit the gas. This comes in the form of something called CTD phosphorylation, which is just a fancy way of saying a chemical modification to the tail of RNA polymerase II. Once that happens, those transcription factors we just talked about release their grip, allowing the enzyme to surge forward along the DNA. We're talking about adding dozens of nucleotides every single second. But the track isn't completely empty. DNA is normally spooled tightly around proteins to save space. So specialized proteins called histone chaperones actually run ahead of our polymerase, temporarily knocking those obstacles out of the way so the machinery doesn't crash.
Now, biology operates on a really rigid traffic system. The polymerase machinery strictly reads the DNA template strand in the 3' to 5' direction, but as it pulls in those complementary RNA nucleotides to build the new strand, it adds them in the exact opposite direction, 5' to 3'. It's this incredibly elegant, continuous process of matching bases just zipping along the track to build a fresh, growing RNA chain.
Which brings us to part 4, stage 3, termination. Our enzyme is moving ridiculously fast, but it doesn't just run off the end of the DNA track randomly. It's actually looking for a highly specific stop sign. Once the specific AU-AA signal sequence, known as a polyadenylation signal, is transcribed, special protein complexes called cleavage factors immediately recognize the code. They latch onto the fresh RNA and physically snip it loose. Now, the molecule released here is called a primary transcript, or pre-mRNA. It's not quite ready for the outside world yet. It's going to require further modifications like splicing, but the core manufacturing phase, that's complete. But wait a second, what about the heavy machinery itself? RNA polymerase II is actually still chugging along the DNA track, even though the main RNA transcript has been cut loose. To actually get the polymerase machinery off the track, we rely on the fascinating torpedo model. Imagine a heat-seeking missile. An enzyme called an exonuclease attaches to the useless trailing piece of RNA still hanging out of the polymerase. It starts aggressively digesting that RNA, climbing up the strand faster than the polymerase is moving, until it literally rams into the polymerase and forcefully knocks it right off the DNA.
It's violent, but man, is it an incredibly effective way to shut the machine down.
So, having seen the intense microscopic regulation required to make just one single transcript, it really begs the question, how does this compare to a much simpler biological workshop? Does every living thing operate this exact same way?
Let's explore that in Part 5, Eukaryotes versus Prokaryotes.
The level of specialization in your cells is just astonishing. For instance, simpler prokaryotes use a single solitary RNA polymerase to handle absolutely everything. But our eukaryotic cells, we utilize 3 distinct polymerases, RNA Pol 1, 2, and 3, and each one has a highly specific job. And then there's the processing. Prokaryotes do little to no processing at all, but eukaryotic cells require 5-capping, 3-polyadenylation, and splicing before the RNA is even allowed to be used. Essentially, eukaryotes have taken a basic manufacturing concept and scaled it up into a highly regulated, multi-layered quality control empire.
The most striking difference to take away from this explainer, though, is the pacing and the location of all this work. Because prokaryotes don't have a nucleus, everything just happens right out in the cytoplasm. They wildly transcribe DNA and translate it into proteins simultaneously. It's totally chaotic. But our eukaryotic cells strictly segregate the work. Because our DNA is locked safely inside the nucleus, transcription has to finish completely before the RNA is sent out to the cytoplasm for translation. It's the ultimate division of labor, perfectly designed to ensure maximum quality control and prevent minor errors from turning into biological disasters.
I want to leave you with this final thought to ponder about the fragile, beautiful precision required to just keep you alive. When you step back and see how highly regulated this system is, from the Tata box recognition all the way to that aggressive torpedo model termination, you realize that every single piece must function perfectly. If just one microscopic mutation broke one part of this assembly line, the entire factory could fail. It's a real testament to the miraculous engineering of the cellular world you carry around inside you every single day. Thanks so much for joining me on this deep dive, and as always, keep questioning the microscopic world around you.